Hi everyone, I am back with our new video, which is about chapter 12, the topic of excretion according to your latest syllabus. So in this syllabus, in this part of the syllabus that you need to know about the definition from this chapter is deamination, which is the removal of the nitrogen containing part of the amino acids to form urea. So you know the amino acid group, which is the NH group actually. So you basically remove the nitrogen containing part to form the urea. So the human excretory system. So try to answer these questions from memory without looking anything up. This will be a good test of your knowledge and understanding of the structure and function of the human excretory system. The diagram shows the human excretory system. Okay, so now let's go to part A, which is label the structure and diagram, which includes aorta, bladder, kidney, renal artery, renal vein, vena cava, urethra, and the ureter. So how can you actually include all these I think it's 1, 2, 3, 4, 5, 6, 7, 8 labelings on this diagram. Okay, the most confusing part is actually the renal artery and the renal vein. Let me just highlight it for you guys. So the most confusing one is renal artery and the renal vein, as well as the urethra and the ureter. These are actually the most confusing ones to remember actually. So how can you actually remember is you just have to memorize the whole diagram. Actually, there's no other option besides that. So the first one being, which is aorta being like the largest oxygenated blood vessel in the whole human body that transport oxygen from the heart to the rest of the body. So this is the aorta. As you can see over here, I will zoom it for you guys. So this is the aorta, as you can see, this arrow coming down. And then the next one is the bladder. So this is the bladder. It's a very simple one, kind of easy to remember actually. The next is kidney, which is, it looks like this, the bean shaped, very easy to remember, bean shaped. And then the renal artery, you can see it over here. This is the renal artery that actually forms from the joining of the aorta blood vessel and it forms the renal artery. So aorta is basically artery, meaning biggest artery of the whole human body that transports oxygenated blood. Okay, so renal artery, from here it goes to the kidney, supplying it with the essential nutrients and the waste products, such as you have glucose, you have I think it's urea, and then you have water, and yeah. And then the next one, which is the renal vein. So you can see this is the renal vein that transports the water, no, that transports the glucose only from the filtration and the ultrafiltration. So this is the labeling of the renal vein over here. It looks like this. As you can see, this is how it is. This is a renal vein, this is a renal artery. Okay, and then in the vena cava, this is the vena cava that transports deoxygenated blood, and renal vein actually joins the vena cava, and this vena cava goes back to the heart. And then the urethra is basically the joining from the bladder to the outer part of the body, and the ureter is where the waste product, or you could say the urea, basically is transported from the kidney to the bladder via this tube called the ureter. So now that we're done with part A, so we can just label it, we're done, and let's go to the next part, which is this. So on the diagram, draw arrows in each of these structures to show the direction in which the liquids inside them flow. So repeat the question again. On the diagram, draw arrows in each of these structures to show the direction in which the liquids inside them flow. So what you have to show, the direction of the renal artery, the renal vein, urethra, and the ureter. So this is how it looks. So this is the direction of the renal artery. You see this arrow? It's referring to the renal artery. And then you have the renal vein. It goes the ways, I should say, after the absorption of the glucose, it comes to the renal vein and it connects the vena cava, which is this. So this is how you label it. And then you have the urethra, which is this, where the urea eventually goes to the exterior of the body. And then you have ureter, which is this. It transports the urea from the kidney to the bladder. Very simple. It's 3 marks actually. You just have to remember the structures where it's located in the human body. So now moving on next. Describe 3 ways. I am sorry for the sound because I live next to a highway, so yeah, it's always noisy. So describe 3 ways in which the liquid contained in the ureter differs from the liquid contained in the renal artery. So what the question actually means, ureter differing from the liquid contained in the renal artery. So the liquid contained in the ureter differing from the renal artery. It's a huge difference, of course, because ureter only contains the waste products, which is the water and the urea, and I think there's another substance I can't remember, but I'll mention it. But in the renal artery, you have like the water, the glucose, you have the urea as well, and one more product actually. So the way how you're going to answer it is going to be like this. The liquid contained in the ureter does not contain red blood cells, white blood cells, or platelets. It contains more urea and less oxygen than the liquid in the renal artery. So actually you guys may have not know about this, as it's not really in the updated syllabus actually, but just for information, and since it's asking the question, the more specific way of answering it's like this. The liquid contained in the ureter, which is the liquid in this part of the tube, okay, it does not contain red blood cells, white blood cells, or the platelets. It doesn't contain any of these, okay. But ureter contains more urea and less oxygen than the liquid in renal artery, because in the renal artery you have all the substances together, the waste products as well as the essential products such as glucose, okay. And however, in the renal artery you only have the water and the urea, and there's obviously less oxygen, because oxygen is needed by the cells of the body, it's needed by the organs of the body, so it won't be filtered out. Okay, so this is about this part of the question, and now that we're done with part C, so let's move on to the next one. So oh yeah, I forgot to mention, usually this kind of question, it's usually worth 2 marks, so I'll mention it here. It's usually worth 2 marks. Yeah. Right. Alright, so let's move on to the next part, which is this. And this is exercise 12.2, dialysis. Let's read this together. In this exercise, you will use your knowledge and understanding of diffusion to work out the answers to questions. If a person's kidney fail, they may be able to use a kidney dialysis machine to carry out the roles normally done by the kidneys. The diagram shows how kidney dialysis can be carried out. So this is actually, I should say, confusing, but it's not confusing, it just need to understand what substances goes in, what substances goes out. Actually, this is like the main thing for you to remember in the dialysis machine. And yeah, so as you can see, fresh dialysis fluid is being put in, and this is the arrow going out, and then This is from where the blood is removed from the vein, and then blood containing waste substances, blood pressure monitor, so that the pressure of the blood is it's going at a constant rate. It doesn't like overflow at a fast speed, or it's not so slow. It's at a suitable rate the blood flows actually. So with this, the blood actually flows through this tube blood pump in order so the purpose of this blood pump is basically in order to regulate the flow of the blood, and the blood pressure is basically to measure the pressure of the blood, whether the pressure of the blood is too high, whether it's too low. And if it's too low, maybe it might need to pump more blood from the vein. And the purpose of blood pump is to make sure it regulates the flow of the blood, so that the blood doesn't like it doesn't stop at one place, because you need a pressure, you need a force to move the blood actually. So then it passes through this dialysis machine, and then with the fresh dialysis fluid, and this part is actually the partially permeable membrane, and then the blood is returned to the body, and then it's back returned to the vein. Now, part A suggests why the blood is withdrawn from a vein rather from artery. So you guys must have understand this from the previous chapter. I think it's chapter 3 or chapter 5, I don't remember. Actually, it's from the heart chapter. That's how I classified by my memory. I call that a heart chapter. So from the heart chapter, it's from the beginning, from the very beginning of the syllabus actually. You need to understand that in an artery there is always a high blood pressure, and since there's a high blood pressure, so if you try to remove blood from there through any tube or wire injection, it's actually going to burst at you. So that's not a feasible thing. But if you do it from a vein, it's actually the blood travels from at a very suitable rate. It's suitable for any person and for any instrument. It's suitable for the instrument to remove that amount of blood or that volume of blood from your hands or from your body, so that it doesn't burst the instrument, or it doesn't it doesn't burst the blood into the person's face actually. Because artery, the blood travels at a very fast speed, and that's like the purpose of artery actually. But in the vein, it's all deoxygenated blood actually, and that is why, since it is deoxygenated, they will take it from a vein actually. So blood in the artery might be, so now in the answers over here you can see blood in the artery might be at too high pressure. There's something wrong with the English over here, but just ignore it. You can later on rephrase in your own words. So basically blood in the artery is at a very high pressure, and it will be pulsing constantly. Okay, it will actually be a better option for the blood to flow smoothly from the patient to the pump, because blood from the vein, it flows at a very softer rate, I should say, a very smoother rate. A better term is a smoother rate, a very smooth rate, and a very I wouldn't say slow, but smooth is the best term for this to describe. And that's why they take the blood from the vein. It can also be dangerous to the patient to have blood taken from an artery actually. Why take it from an artery? Because it's not just that the blood is going to burst the instrument. It can actually you could say it could actually harm the flow of the blood in the artery that's normally traveling, because you're just like how something is traveling at a very fast speed, and then all of a sudden there is like an obstruction along the way. So that can actually harm the flow of blood reaching the cells of the body, reaching the organs of the body that actually need that oxygenated blood. Alright, and this kind of question, it's just worth 1 mark only. Usually 1 mark looking at the number of lines provided to you. So I'll just mention it here. It's usually a 1 mark question. So suggest why the blood is withdrawn from a vein rather than from an artery. So you can say blood in the artery might be at too high pressure and it will be pulsing. Another option, I should just make it like a slash or something. It would be better for the blood to flow smoothly from the patient to the pump. Okay. I'm sorry. Okay. It might also be dangerous to the patient to have blood taken from artery. Why dangerous? Because it's having a high pressure and pulsing, and if it's taken from the vein, the blood flows smoothly from the patient to the pump. So you want a smooth process. You don't want a very hectic process of the transport of the blood. Alright, so now we're done with part A, so let's highlight that first actually. So it's actually a practice of mine. Whenever you're done with whenever you're done answering any question, just highlight that part of the question so that you know you've completed that and you're sure of your answer. So now let's move on to part B. Suggest why the blood flows through many small channels in the dialysis unit rather than through a single large channel. So what they're actually referring to, the many small channels, is actually they're referring to these very small channels rather than through a lot, because it increases the surface area across which diffusion can take place, it speeds up the process. Exactly. So since a dialysis machine and a person needs to a person whose kidney is not functioning well, so the person obviously needs a dialysis machine. If the person cannot afford the the kidney transplant from the donor, or there is some like autoimmune response with the rejection of the foreign kidney, in simple words, so then there's no other option for the person but to actually opt for the option of doing the dialysis option actually. So with this, the reason for having many small chambers is because when there is a large, so since you need to understand that if there is a large surface area, that means there is more space for the molecules to collide with each other, and when there is more space, when there is more surface area for the molecules to collide, that means it's going to speed up the diffusion process between the partially permeable membrane that's present in the dialysis machine, which is usually the thing that's always kept constant is the glucose. The glucose is kept constant because the person needs glucose in the body. You don't remove glucose because glucose is not a waste product. So the fresh dialysis fluid that's put in, it's actually the glucose that's actually at the same concentration as the glucose concentration in the human body. Okay, and since the waste product is actually going to come from the way into the dialysis machine, so glucose is also present in the vein as well, in the fluid. Okay, so since the concentration is going to be the same, but there's a large surface area, there is no diffusion that's going to take place among the for the glucose. But for other substances, like you have salt, you have urea, these two things, the concentration is not the same. Like the dialysis fluid that's put in the machine, that concentration of that is lower, or it's just not the same as the concentration of the salt and urea taken from the person. The reason behind is because the waste product includes the salt and the urea, so these are like important to remove from the body, or else it can cause, since they're very toxic, can cause harm to the organs actually. So as you know, if there's a large surface area present, there'll be more space for the molecules to collide. If there's more space, diffusion, there'll be more chances of diffusion to take place, higher chance of diffusion. Diffusion is what? From an area of high concentration to an area of lower concentration. So with that, it's actually going to speed up the whole process. So why the blood flows through many small channels? So you need to understand, when there is an increase in the surface area, the look of the picture, or increase of surface area, meaning it's actually going to be thin. It's going to actually be very thin actually. So that is why they include small channels in the data. These are the small channels, and as you can see, they're actually thin rather than through a single large channel. So when you have these small channels, it actually increases the reliability of making sure that all these substances, they are removed and the essential ones are still remained in the fluid, like glucose is remained actually. So the main reason for you to explain, because of the presence of the small channels in the dialysis unit, which are these, as you can see over here, it makes for the process of the diffusion to occur, and it thus speeds up the process. And this is usually worth about 3 marks. Yeah, it's usually about 3 marks actually. Don't be surprised, but it's actually about 3 marks. Yeah. So let's move on to the next question. The table shows the concentration of some of the substances in the dialysis fluid and in a person's blood plasma before it enters the dialysis fluid. Okay, so substance glucose, concentrated dialysis fluid 5, kind of blood plasma is 5. See, glucose remains the same, because you don't have to remove glucose, because glucose is not a waste product. It's needed by the human body, so you don't remove that. But since it flows along in the blood, you can't remove it, so you have to keep the concentration the same. Protein in dialysis fluid 0, but protein blood plasma is 8. Okay, take note of this. Urea in the dialysis is 0, but concentration blood plasma of the urea is 7. Use the information table and your knowledge of how dialysis fluid takes place. Okay, so right, so what will happen to the concentration of the glucose in the person's blood as it passes through the dialysis unit? So now they're asking about the concentration of glucose in the person's blood as it passes through the dialysis unit. I don't know how you handle that, but yeah, this is how it is. So now explain your answer. So the concentration of glucose will actually remain unchanged, and as there is no diffusion gradient for it, and the number of glucose molecules moving in each direction will roughly be equal as well. So as I mentioned earlier in the video, that there are like 3 or 4 essential substances. There are actually 3 or 4 essential substances that has to pass through the dialysis machine, but the only thing that will not diffuse out is glucose. Glucose will remain the same. And since the question is asking about the concentration of glucose in the person's blood, so the concentration of glucose in the person's blood actually will remain unchanged, because there is no diffusion gradient that's been created in the dialysis unit for the glucose, so nothing will happen to the glucose. It will not diffuse, and the glucose molecules in the fluid of the person's blood, as well as the fluid in the dialysis machine, the molecules of the glucose will actually be equal almost. So now that we're done with this part of questions, so let's highlight this. Now the next part: what will happen to the concentration of protein in the person's blood as it passes through the dialysis unit? So what happened to the concentration of the protein in the person's blood as it passes? As you can see, the concentration in the dialysis fluid is actually 0, but the concentrated blood plasma is actually 8. So of course there is a diffusion gradient that's been created. So you will say that the you won't really say that, you will think that there will definitely be a concentration gradient, because over here the dialysis fluid is 0 and this one is 8. You automatically think that of course there'll be a diffusion gradient in the dialysis unit, since the concentration of fluid is 0 and the concentrated blood plasma is 8. but you need to understand this is a very tricky question. Protein cannot be diffused out of this small unit of channels. Why is it that? Is because the molecule of protein itself is just so big. Okay, the molecules are just too large, okay, to get through the holes in the dialysis tubing, so they'll all stay in the blood actually, and hence it will remain unchanged, just like the glucose. So don't be confused just because 0 here is 8 here, so definitely it's going to have a concentration gradient. No, it doesn't happen that way. If you think carefully, there are 4 essential substances that pass through the kidney. The first, like you need to understand, generally there are actually 4 substances that pass through the kidney. The first one is water, you have salt, you have glucose, and you have urea. Okay, but in the table over here, what they only have given you is actually glucose and protein and the urea. So by looking at this value over here, you guys will definitely think that, oh, this is definitely going to concentrate. But you need to understand, protein, since the size of the molecule itself is so big, it's next to impossible for protein to actually pass through the dialysis tubing. It's next to impossible for diffusion to take place actually. So nothing will happen, the concentration of protein percents, but it will remain unchanged, or you can say it will remain the same. It's the same thing, and they'll all stay in the blood. Okay, so don't be confused, because protein is not part of the filtration process, so remember that. All right, so now that we're done with part D, let's move on to part E. So what will happen to the concentration of urea in the person's blood as it passes through dialysis units? So now we all know that urea is actually a waste product, and since it's a waste product, it's not good for the human body. So definitely the first thing that will come in your mind is that it has to be removed. So since it has to be removed, there's a concentration gradient. So since there's a concentration gradient, that means diffusion is going to take place. So in the explanation, the way how you write is that the concentration of urea in the blood will actually fall. Why? Because the higher the concentration of urea in the blood than dialysis fluid, so it will diffuse down its concentration gradient. It will diffuse down, okay, through the dialysis membrane. All right, so you need to remember this. What will happen to the concentration of the urea in the person's blood as it passes through the dialysis unit? So explain analysis. So you could say the concentration of urea in the blood will fall. So you need to understand this concept. It's very important, because if you don't understand what is actually decreasing, what's actually being the same, it's difficult for you to answer this question actually. Okay, so you need to know that urea is not wanted by the organs, it's not wanted by the cells of the body, so it definitely has to be removed from the body. So it's going to be diffused, and we know that in the person's fluid in the dialysis machine, the concentration of urea is going to be very high, and the concentration of the urea in the dialysis fluid is purposely kept on 0. Okay, why it's purposely kept on 0? So that there is a diffusion gradient that's been created, so that the concentration of urea in the blood plasma can diffuse from a higher concentration to a lower concentration across a partially permeable membrane. Okay, it will diffuse down through the dialysis membrane. So this is how you explain the answer. So I actually forgot to mention like how many marks are these actually worth of. So I'll mention it right now here. So the concentration of glucose will remain unchanged. So this is roughly about 2 marks. You can say 2 marks. And this one, the protein will remain unchanged. This is also roughly about 3 marks actually. Why is it 3 marks? Because the first point I mentioned will remain unchanged. The second point, molecules are too large, so it can't get through tubing. And the third point, it will stay in the blood. So these are the 3 points you need to state. Since there are 3 lines, so 3 points. But in this case, looking at this, it's actually usually, you can say it's 2 to 3 marks for this question. Actually, it's 2 to 3 marks, because the first point, as you mentioned, it remains unchanged, and the second point is, since there is no diffusion gradient, It could be, but I would say it's 2 marks, but it's up to you. So what will happen to the considered urea in person's blood? So urea concentration in blood plasma is high, but considered urinalysis it's 0, so you have to mention it's roughly about 3 marks as well over here. So yeah, this is how it is. And now that we are done with this chapter of the IGCSE Biology Workbook 3rd edition. And if you either like the video, do like and subscribe. And if you guys think that there is some error in explanation or I wrote something wrong over here, you can mention that in the comments section below so that I can pin your comment if I deemed it suitable to pin it. And yeah, that's all for now, and stay tuned. Don't forget to like and subscribe as it means a lot, and you guys could continue to receive amazing content. So see you then. Alright, bye.